So hi everyone, I'm gonna discuss some structural insights of a protein called MECP2 using AlphaFold, and specifically CpG binding and its effect of function by phosphorylation. And I would also like to thank my mentor, Dr. Jody Krause from Princeton University. So just a brief overview. I'm going to talk about basically what this protein is, the methodology I used for this research, what it means to be CpG binding, and how phosphorylation affects it, and the possible implications. So here is a picture of the methyl binding domain of this protein, bind it to DNA. So you can see over there, that green thing. And so I'm going to introduce you to epigenetic modifications, which includes DNA methylation. And so it plays a crucial role in gene expression. So the MECP2 protein, which is the methyl CpG binding protein, is a key mediator in the process, as the CpG island is a region where DNA is methylated, has a methylated cytosine, and that is followed by a guanine. So the MECP2 protein can recognize the methylation at the 5th carbon of the cytosine, as you can see labeled here. You can see there's a slight difference between these two things. And they can recognize it and subsequently alter the chromatin structure, which affects gene expression. So there are many implications of how epigenetics may affect health, human health, including cancer, metabolics, and we will see how this is particularly important in my research, where it's important in the nervous system. Okay, that's fine. So Rett syndrome is a rare neurological development disorder that primarily affects girls because the gene MECP2 is found on the X chromosome. And the reason for that is actually most of the boys who have this defected version of MECP2 almost always just die out because they only have one X chromosome and there's no way to mediate for that. So affected children typically develop normally for the first 6 to 18 months before entering a period of regression, which they will lose their motor and communication skills. They have loss of purposeful hand movements. As you can see here, I didn't mean to hide it, but repetitive hand movements are flapping and clasping hands, and worse symptoms include like seizures. There's also evidence to suggest that phosphorylation of MECP2 can decrease its affinity to bind to the CpG island of the AVPG, leading to the hyperactivation of this, which is the hypothalamus pituitary adrenal axis, so HPA in short. And so as you can see from this diagram, well, you can't really see. Well, as AVP is increased, the level increases. It stimulates the release of ACTH, which in turn releases cortisol into the immune system, which is a stress hormone. Which is a stress hormone, and so that is thought to contribute to depressive disorders. So as for Rett syndrome, currently there is no cure and only supportive treatments such as like physio and speech therapy. So there are various sites where MECP2 is phosphorylated, as seen in this picture. It decreases MECP2's, so thought to affect MECP2 function. In the MECP2 E2 isoform, it includes serine 86, serine 164, serine 274, etc., and generally associated with decreased DNA binding. So it's essentially, with the phosphorylation of certain residues, it binds less well to the DNA, while dephosphorylation at S80 is thought to promote chromatin dissociation. So this study gives the residue numbers, however, on the E1 isoform. So as there are similar proteins, but they occur differently in the human body. So the E1 isoform is more expressed in the brain, so which is what we're curious in. However, the methyl binding domain in both of the isoforms are identical, so I can take a look at that, and it doesn't matter which protein it really is. So yeah, so what I've used is a software, or a server called AlphaFold, which is a protein prediction software. As some of you may know, it has won last year's Nobel Prize, so it would be one of the more trendy topics. And so this study uses AlphaFold and PyMol to predict and analyze the structure of the human MECP2E1 isoform. And the methylated DNA molecule used in the structural part is the BDNF gene, which is called brain-derived neurotrophic factor, which is crucial for development of nerve cells. And I use this because I wanted to compare it to a protein databank structure, which is an experimental structure, a known correct structure, and which has this section of the gene. Now, a somewhat interesting thing about a CpG island is that it's palindromic. So if you read it forwards and backwards, from 5' to 3' on both strands, it both goes C and then G, and C and then G, which is quite interesting, I guess. And then, so that's quite key to how the protein recognizes it, because it needs to have 2 cytosines, 2 guanines. That has both methylated cytosine. And so what I did is I posted a section of the fastest sequence, which is the amino acid sequence of the protein, into AlphaFold. And I've chosen, since AlphaFold now has a post-translational modification function, I can choose to phosphorylate one of the serine residues and see what the structure arises. And yeah, so that is what I did. So here's the first thing I've tried, which is I've pasted the whole amino acid sequence of the MECP2E1 isoform. And what it got is large regions of disorder that is, well, not really helpful to the experiment. Because as you can see, these coils, they are completely not correct. So this is because, as AlphaFold, it is a pattern recognition, essentially, sort of server. It doesn't actually understand how the protein chemistry actually would be, but instead it just recognizes patterns. And as you can see, the blue part is the actual binding domain of this protein, so it binds to the DNA. And so you can see that is pretty ordered. As you can see, it's labeled in blue, which tells me that, and it has higher reliability. So there are several factors that indicate this. So first, the PLDDT score from 0 to 100. It tells me how reliable or how confident it is in its prediction, as well as PTM score from 0 to 1, which shows the overall accuracy, and IPTM, which shows the accuracy of the complex. So a protein that is attached to something else. So in this case, DNA. So that's a complex, and it reflects how confident it thinks it is. And so the methyl binding domain, we can see it predicts in a fairly high confidence. Here's just a side note, because as you can see, it's a slightly different structure, but this was done around a year ago, and this was very recently. So we can see that this technology is evolving, and they're changing it to update it. So here's just showing you the sequence that I have used to make this sort of prediction. And here's the MBD, specific sequence of the methyl binding domain, and the BDNF gene CpG island. So here's a UI of, the software I used, PyMol, which I can identify specific amino acid residues in the overall protein, which is useful. So there's also something known as RMSD values of AlphaFold. So RMSD values is essentially like shows how similar two structures are, measuring average distance between two structures, in this case in angstroms. So as you can see in all the predictions, comparing to a real structure, which is a protein data bank structure that someone had to experimentally obtain, they're both fairly similar. So it's showing that the AlphaFold did a good job in predicting the following structures. So by looking at the protein structure using PyMol, we can see several things. So first of all, as I've mentioned to you, the CPG island, it's palindromic. So there are two guanine residues. As you can see, this chunk is guanine, and what I've labeled R is arginine, which is an amino acid residue. And what happens is they form intermolecular forces between them and guanine, which is one of the ways it binds to that specific part of the DNA. As you can see, arginine 123 and arginine 145 in this case, and as well as serine being electrostatically attracted to the phosphate groups of the cytosine nucleotide, which is how it binds to it. And thirdly, which is more interesting, is that around the cytosine, methylated cytosine, is a hydration shell. So that means that water accumulates around the methyl group. And that's interesting because methyl groups tend to repel water. So what people have thought throughout the years is that there will be some hydrophobic interactions between the amino acid residues and the DNA. However, what actually happens is that the amino acid residues actually interact with the water surrounding the methyl group, which is also a key way that it interacts with this part of the DNA. And so these three ways are key for me to understand like how the changes in the protein where it has been phosphorylated can actually change its structure. So, sorry, just to go back. The hydration shell can potentially explain why residues, the tyrosine 135 residue, when it becomes phosphorylated, it's found in many Rett syndrome patients. And you can see it's possibly to do with the changes in its interaction with the water shell, the hydration shell around the methyl group. So here's some things I have chosen to change in terms of the protein. I have made some post-translation modifications which AlphaFold supports. So that includes phosphorylation of serine 125 and serine 146. As you can see, there's repulsion between the phosphate ions when serine 125 is phosphorylated, which confirms the hypothesis that the affinity to DNA is decreased. However, the second one, it shows that it actually bonds covalently to the phosphate group. And that is it should just simply be one of the limitations that AlphaFold hold, that it does not really understand like chemical properties or stability, because these bond angles should not be very stable. And therefore, this should not be very feasible, but it's just one of the limitations that it forms a little triangle. And second thing I sort of did was serine 98 and serine 176 phosphorylation. So these two don't directly affect binding between the protein and DNA. However, it makes a conformational change. And what happens is that when you change something of the protein at one end, a separate end could also experience changes. That's called allosteric change. And so what happens here is that the arginine residues of the protein interact with the phosphate groups attached to the serine. And this leads to the loss of attraction between the arginine and the guanine group, as well as serine and the phosphate group, which again confirms the hypothesis that there's some sort of decrease in affinity due to the conformational change. And lastly, it's just a separate thing, serine 128 phosphorylation. So there's also salt bridge interactions or ionic interactions between residues and phosphate groups, which can lead to, again, disruption of arginine 123 binding with guanine. And yes. So lastly, I want to talk about just a bit about HDAC, which is histone deacetylase. So it's usually, by removing acetyl groups on histones, which are proteins that DNA attach to, it promotes gene expression. So HDAC also interacts with MECP2 as proteins form complexes and they interact with each other. However, the protein has something called the TRD, which is transcription regulatory domain. And as you can see, this is the disordered part of the protein that I was talking about. So I was trying to see if by adding HDAC, it would sort of fix it into stability, because it's what meant to happen. They're meant to interact. However, this was not the case. It was still very messy, and I could not see what was going on. However, the methyl binding domain did show interactions between HDAC and MECP2, but there's no way to know whether that is intended because there's no experimental structure, and so you cannot trust AlphaFold on this. As well as there's the obvious limitation that DNA exists in euchromatin and heterochromatin, and there's histone proteins and all other kinds of proteins that HDACs interact with, so the predictions were in the end unsuccessful. So there are some possible implications of this study. So here's like a list of modifications I played with and their interactions with the DNA structure. So it's worth noting that the typical hydrogen bond distance is 2.2 to 2.5 angstroms, which shows that if you look in the previous models, that the loss of affinity is clearly shown because it's exceeded this number. And so is there a possible way to target some of the depressive disorders or Rett syndrome that seems to be caused by phosphorylation of MECP2? So first of all, there's a kinase protein which adds phosphate groups to proteins. And the problem with that is if you inhibit it, it's not a specific protein. It can target all around the body. So that's a question to ask, whether we can find a specific inhibitor to just inhibit a certain group of kinases. And there is what is known as the KI-PIK, which can produce a fingerprint for the identification of kinase acting on targeted phosphorylation sites. So that's very helpful if we know which phosphorylation sites causes disease. And lastly, future work, I would love to work on the hydration shell of around the MECP2 and CPG island. However, AlphaFold does not support that, and obviously shows that we still need humans to do our experiments to see that what's actually going on, which is still very interesting. And that is the end. Thank you.
Thank you, Felix, for a wonderful presentation and a really good one in terms of its implications and future directions. Is there any questions to start with?
Yeah.
How did you choose to focus on ECP2?
Yeah. So I read a book. It's called Epigenetics Revolution by Nessa Carey. And it has the protein in it and talks about the phosphorylation of this protein particularly, but it only mentioned it in like one sentence. So I thought it was interesting, and I just looked into it. Yeah.
A quick question from my end.
Yeah.
Can you give us a bit more info on how much time the simulation takes? Because I feel that's one of the major advantages of the study and the major interesting thing about AlphaFold is super fast.
Yeah.
Since you kind of ended with we need humans, probably we need to give
Yeah.
a little bit info on the advantages of AlphaFold for this project.
Yeah, there's countless advantages of AlphaFold. If I could just go back to this, it's very beginner-friendly software. You can, most people who study just a bit of biology or chemistry can know like the four DNA bases, and you can just paste that in, and it'll give it to you. You can make modifications if you click this button, and it gives you the structure within let's say maximum 10 minutes. So it's really fun to work with, and yeah.
10 minutes is a really significant time cutting, because if you show us one of the DNA, the other slide, this one, I think we are looking at about 2,000 to 3,000 atoms. Each of the string, the bend or itself, they're one of the atoms.
Yeah.
So if you want to actually simulate, each atom interacts with other 2,999.
Yeah.
So I do pure chemistry studies, as you mentioned, it takes about 15 days if they run it in a supercomputer in Cambridge. So 10 minutes is really approximation. And but it shows really good results. I think with the combination of AlphaFold and further studies, it eliminates the need to do a lot of actual calculation. Like the ones you found, there are a lot of possibilities of different phosphates and different binding sites. If you kind of give some of the conclusion, like possible binding and possible cut down on things, the next steps it's going to be easier to focus more on.
Yeah, I think for sure it's a combination of human and
But a really fun start. Thank you. Any other questions? If not, I'll end with one question. What are the next directions you're looking at? Would you like to explore more proteins, binding sites, or different? You mentioned about shells, right?
Yeah, I think that one's interesting. I don't know how much I can do with AlphaFold though, because it relies on like an experimental thing. It's like there's a hydration shell around this DNA that the protein seems to be interacting with, but I guess I could sort of directly compare that to the AlphaFold version to an experimental and see the distances between water molecules, but doesn't seem to be like very nuanced.
No, that sounds good.
Yeah.
RMST studies are really good. One other question is, you mentioned about the root mean squared error, that stands for RMST, but the values are somewhere between 0.5 to 1.1. Is it a scaled value or?
It's the average, it's the average distance between each residue in Armstrongs.
Did you scale it back to 0 to 1? Because I can see 1.1.
No, it's not scaled.
So
because
is it a good or a bad value? 0.5 means?
It's a good value. So I think 2 is a good value. So below that, it shows it's similar. Yeah. So AlphaFold is confident in that and the experimental structure. Yeah.
So the final thing for your future suggestion is, I think the things you're looking at is hydrogen bonding and van der Waals force. CH3 as the CH bond, and you see the interaction with hydrogen bonds.
Yeah.
So it's one of the most challenging things to do because it's very weak interactions.
Yeah. No, but yeah.
But it's good that you are trying, because it's one of the challenging things, and I think there are a lot of scopes out there.
Yeah.
You can help the other people like us to save time.
It's not. I think directly interacting with the CH3. Instead, it's other parts, but as somewhere.
I thought it's H interacting with the CH bond, isn't it?
Yeah, but
it's hydrogen bonding and van der Waals.
But not with the actual CH3. It's so this can bond with the water molecules, but in other cases the arginine interacts with the guanine, which would be a hydrogen bond. Different.